So I am Biswanath, and today we are going to read the medical genetics, which is given in your CBME curriculum. Okay, so medical genetics is an important part of your anatomy curriculum. Sometimes question may come on the disorders. You can read this chapter from the standard textbooks also, but you don't have much time to devote to medical genetics. Of course, genetics is a huge chapter. Okay, some part of genetics you read in biochemistry also, and some part you have to read in your anatomy. So we are covering the genetics of the anatomy. Okay, let's start. Genetics. So genetics is a branch of biology which deals with inheritance. Okay, which deals with inheritance. So inheritance is the process by which the children acquire characteristics, the traits. The traits are acquired by the children. Okay, it's an inheritance. Okay, and the genetics is related to the genes. The genes is the basic of genetics. Okay, so gene is a hereditary unit. It's a hereditary unit. It is a segment of DNA. This segment of your DNA. Okay, and inheritance is the process. Inheritance I told. Okay, there are about 50,000 to 1 lakh genes per cell. Okay, so each gene has 3 components. Okay, each gene has 3 components. A gene has 3 components. One is your deoxyribose sugar, your nitrogenous base, and your phosphate group. Okay, and each gene has 2 parts. Each gene has 2 parts. One is called your exon, okay, which is functional, which expresses, and another is called your intron, which is the silent part. Okay, then there are types of genes. Okay, there are many types of genes. Types of genes are there. Okay, so there are regulatory genes. Okay, then there are structural genes. Structural genes are basically producing the proteins. Okay, like Z, Y, and A genes. Okay, in the lactose operon. Okay, Z produces your beta galactoside. Okay, A produces your permease. Okay, and A produces your transacetylase. Okay, so these are the structural genes. Then there are operator genes. And other types of genes are dominant and recessive genes. Okay, dominant genes are those which can express, okay, whether the genotype is allelic, like homozygous, or heterozygous. Like in plants we have AA. Let a gene be heterozygous. This is heterozygous. Okay, and this one AA is homozygous, but these both are dominant genes. Okay, so this can express. And then we have recessive genes which can't express, like AA. It will not express. Okay, understood. Then there are co-dominant genes also, which are exceptions to your Mendelic inheritance formats. Okay, some are co-dominant genes. Okay, like AB. This will also express. This A and B both will express. Okay, they express concurrently. So codominant genes express concurrently. Understood? Then some are called as sex linked genes and some are sex linked genes. and your sex limited genes. Okay, so what are sex linked genes and what are sex limited genes? Let me tell you. So sex linked genes are genes, okay, which are located on your X chromosome or your Y chromosome. Okay, these sex linked genes are present either on your X chromosome or your Y chromosome. Okay, and they're inherited differently between males and females. Okay, sex limited genes express in only one sex, express in either male or your female, only one. Okay, like example is gene for pattern blindness, alopecia, alopecia, alopecia gene, this one. Okay, and carrier genes are heterogeneous recessive genes, heterogeneous, and this is a note point, heterogeneous recessive genes like AA is a carrier gene. Understood? Let's talk about alleles. So alleles are basically each diploid cell has a pair of genes, okay, for each characteristic or traits, which is called as alleles. Okay, for like hair color, okay, for like hair color, for black hair color as well as for gray hair color. Okay, these are called traits, but the genes encoding the black and the gray, these genes, this alternate form of genes, okay, alternate forms of genes are called as alleles. Understood? Okay, if the two alleles are identical, they are called as homozygous. Okay, and if two alleles are not identical, different, two alleles different, heterozygous. Okay, homozygous and heterozygous for that particular trait. Okay, usually what happens that one allele expresses, which is called dominant allele, and one allele does not express, which is called as recessive allele. Okay, so genotype is the genetic makeup of an organism. Okay, it is called as genotype. So genotype is of three types, like homozygous dominant, okay, then your homozygous recessive, and your heterozygous. These are genotypes. Homozygous dominant like EE, okay, and homozygous recessive like EE, and heterozygous like EE. Okay, heterozygous like EE. So here the phenotypes, phenotype is the external appearance that arises due to the genotype. The homozygous dominant and the heterozygous, these have same phenotype. These will have same phenotype. Okay, and this will have another phenotype. This heterozygous recessive will have another phenotype. Okay, then let's sometimes a short note on your bar body or your sex chromatin is asked. So let's learn about bar body. So what do you mean by the bar body? Bar body is the sex chromatin. Females have XX. Okay, this is a females. In females, inactivated chromosome, okay, this chromosome, let's say, is inactivated chromosome, inactivated X chromosome, okay, which is attached to nuclear membrane of somatic cells, attached to nuclear membrane of somatic cells. Okay, and when it is inactivated, okay, inactivated during second week of gestation. Okay, it was first noticed by Barr and Bertram in 1949. Okay, Barr and Bertram in 1949. It is inactivated during second week of gestation. Okay, and the cells with Barr, how to calculate the number of Barr bodies? Okay, so Barr body is equal to number of X chromosome minus 1. Okay, so in normal female, one Barr body is there. Normal female, one Barr body is there. In your Turner syndrome, where there is XO genotype, there is no Barr body, as x 0 minus 1 is 1, 1 minus 1 is 0. Okay, and your Klinefelter's syndrome, where there is triple X, XXY, XXX, okay, or your multi X female, so there is 3 minus 1 is 2 Barr bodies. Okay, so now you can calculate the number of Barr bodies too. Okay, and you can write a short note on Barr body. The cells with Barr body are chromatin positive, and cells without Barr body is chromatin negative. Another thing you can write is some morphological features of the Barr bodies. Okay, so Barr body is a darkly stained part, plano-convex shaped, and about 1 μm in diameter. Okay, understood? So you can deal the Barr body questions. Okay, if it is a short note, most probably it is a short note. Okay, and other related questions related to Barr body, if there's fill in the blanks or so. Okay, then let's write about Lyon's hypothesis. Let's discuss the Lyon's hypothesis. So what is Lyon's hypothesis? Lyon's hypothesis means the number of Barr bodies is 1 less than the number of X chromosomes. Okay, the same thing which I wrote there was the Lyon's hypothesis. That means the number of Barr bodies is n minus 1, well, n is the number of X chromosomes. Understood? So in Klinefelter's we have 1 Barr body. Okay, so here I did a mistake. Okay, this is got confused in Klinefelter and multi-X female. No problem in this Klinefelter syndrome. It is XXY. Okay, so there I was just wondering something else and writing something else. No problem. 2 minus 1, that is 1 Barr body. Okay, any other things you can write, like multi-X female, like XXX, that is 2 Barr bodies. Okay, understood? You can write this, and this statement is called as your Lyon's hypothesis. Just remember this. Now let's talk a little bit about chromosomes. Okay, chromosomes. So talking about chromosomes are darkly stained structures. They are darkly stained, rod-like structures darkly stained rod like structures present in the nucleus of the cell, okay, formed by condensation of chromatin, your chromatin. Understood? So each chromosome has identical filaments called as your chromatids, okay, and they are joined together at a constriction called a primary constriction. Okay, let me draw the structure. So here we have the chromosome, okay, so this is the long arm, and here we have the short arm, okay, long arm and the short arm, and here you have two satellites present here, two satellites, okay. Then in the satellite there is a constriction, you can observe here, this one, which is called as a secondary constriction, and this one is called as a primary constriction or a centromere, okay, and there are two discs attached to it, okay, there are two discs attached to it, discs which are called as kinetocores, okay. The free ends are called as your telomeres, okay, the free ends of your chromatin are called as your telomeres, okay. Understood? So there are types of chromosomes also, okay, types based on position of centromere, okay, types based on the position of centromere. We can have many types of chromosomes, basically 4 major types of chromosomes, okay, you can have 4 major types of chromosomes. So it can be metacentric chromosome, okay. In metacentric, both arms are of equal length, okay, both arms are of equal length. This classification is also called as a Denver's classification. If I just want to say more, Denver's classification. Look, genetics have a wide syllabus, okay, it has no defined syllabus. I can teach more and more, okay, and you can learn more and more, but I am teaching the pinpoint things which you need to understand and write for your university exams or NEET PG entrances, okay. So for NEET PG you can go a little bit advanced also, and you can research and practice MCQs and add points from them also, okay. But I'm teaching the bare minimum of genetics which you need to know in anatomy, as per anatomy is concerned, okay. You can read genetics in your physiology or your biochemistry, or you can purchase a textbook, okay, for genetics as a whole. Textbooks is there, like GK Pal genetics is there, okay, GK Pal genetics is followed, okay, and you can buy the gold standard, okay, the gold standard for genetics, that is Emery's principles of genetics, okay, Emery's principles of genetics. You can buy the gold standard textbook also, okay. I'm teaching just the bare minimum things which you need to know, okay. And then we have submetacentric chromosomes, submetacentric chromosome, which is close to the midline, okay, just close to the midline, that means like this, something like this, okay. This arm is little bit longer than the upper arm, okay. Then we have acrocentric, and then we can have telocentric, okay. Acrocentric means you can just observe like this, and telocentric is no shorter, just like this, okay. This is telocentric. It is not found in humans. This is not found in your humans. Okay, so these are your types of chromosomes that are present. Okay, these are the types of chromosomes that are present. It was also given in your NCRT also. Okay, so let me share some extra point, that is short arm. This short arm is also called as a P arm, and your long arm is also called as a Q arm. Okay, just an extra fact just to do. Okay, so PQ ratio is sometimes asked. Okay, PQ ratio. So for telocentric, PQ ratio is your short arm by long arm, and short arm is 0. Okay, 0 in length, by Q is 0. So for telocentric chromosome it is it is about 0. Okay, in acrocentric it is likely to be 0. In submetacentric it is less than 1, and is metacentric, P by Q is equal to 1. Okay, so P by Q ratios are also asked somewhere. Okay, then we will learn about something about karyotyping. Okay, then we'll talk about karyotyping. Let's learn about karyotyping. Okay, so karyotyping, what do you mean by karyotyping? It is the process of studying the chromosomal pattern. Okay, we study the chromosomal pattern. Okay, it is done by arranging the chromosomes in groups in descending order of their lengths. Okay, like pair 1 2, we go to the all the autosomes, we arrange the autosomes up to pair 22. Okay, pair 22, and the pair 1 is the longest. Okay, and the pair 22 is the shortest, and it is arranged in 7 groups. Okay, is arranged in 7 groups. Okay, 7 groups. It has denoted from A to G. From A to G there are 7 groups of chromosomes. Okay, and karyotyping is helpful in determination of, it is important in determination of sex in your ambiguity. If there is ambiguous sex, or some organs are not yet developed, the genital organs, then it is helpful, karyotyping is useful. Okay, and the aberrations, chromosomal aberrations. Okay, understood? Okay, so after that if you want to see the karyotype of a normal male. Okay, so I'm bringing a picture here. Okay, so here are we, this is a karyotype of a normal male. Okay, so this is a karyotype of a normal male. Okay, karyotype of a normal male. So if you observe 1, 2 and 3, 1, 2 and 3 are nearly metacentric chromosomes. Okay, 1, 2 and 3, these 3 chromosomes. Okay, these are long and these are metacentric in nature. Okay, then we have 4 and 5, which are also long but submetacentric. 4 and 5 are long but submetacentric. Okay, 6 to 12 are medium sized and also submetacentric, starting from 6. Okay, to all of this, these are submetacentric plus medium sized. Okay, and from 13 to 15, this one, these are 13 to 15, these are medium sized and acrocentric. Medium sized and acrocentric. Okay, 13 to 15 are medium sized and acrocentric. From 16 to 18 there is short and submetacentric. You can see the size decreasing, short, and your submetacentric. Okay, 19, 20, 21 they are short and metacentric because they are very small, so the shape nearly becomes metacentric. From 19 to 20 these both are what? Short and metacentric. Okay, and 21 and 22 are very short with acrocentric. 21 and 22 are very short with acrocentric. This both very short and acrocentric. Okay, so as it is a normal mean, so xy must be present. Okay, understood? So these are the karyotyping. This is the karyotyping. Okay, so now we go to an important topic which is called as your karyotyping. Okay, sorry, not the karyotyping, chromosomal aberrations. Chromosomal aberrations. Chromosomal aberration. Okay, so in chromosomal aberrations they include either changes in either structural component or the number of chromosomes. So structure or the number of chromosomes, this is altered. Okay, this is altered. Understood? The chromosomal aberrations may be divided into structural aberrations or they may be numerical aberrations. Numerical aberration. Okay, structural aberrations, like many types are there. We discussed three important types: deletion, microdeletion, and leukemia. Not leukemia, the aberration is called as your translocation. Okay, leukemia is the disorder. Okay, translocation. Okay, deletion, for example, on the fifth chromosome. Okay, fifth chromosome, the short arm, the fifth chromosome is deleted, is cut in a syndrome called as your Cray-Duchat syndrome. Cray-Duchat syndrome or Cat's-Cry syndrome. Okay, then the microdeletion part. Microdeletion is seen in the Prader-Willi syndrome. Prader-Willi syndrome. And translocation is seen in leukemias. Like your Philadelphia chromosome. Okay, and chronic myelogenous leukemia and all. Okay, we'll discuss about that. Numerical aberrations may be basically of two types. One is aneuploidy, another one is polyploidy. Okay, aneuploidy is just the addition of one chromosome, or addition or deletion of just one chromosome. Okay, but polyploidy is the whole set of chromosome, addition or deletion of only one chromosome, but this is the addition or deletion of a set of chromosome. Understood? So aneuploidy may be trisomy or monosomy. Trisomy, as in Down syndrome, trisomy 21, we will discuss about that. 2n plus 1 condition. And the monosomy, that is 2n minus 1 condition. Okay, and polyploidy may be triploidy, diploid or normal, triploidy, and tetraploidy. These are important. Polyploidy, in polyploidy. Okay, so chromosomal aberrations, just look at this. Okay, these are the classification of chromosomal aberrations. Let's discuss about translocation. Okay, translocation is an important short note. So translocation is a structural aberration of chromosomes. A structural aberration. Okay, so there is a breakup chromatin of one chromosome, so it becomes fixed to the second chromosome. Okay, so like if here is a chromosome, okay, if here is a chromosome and here is another chromosome, okay, if there is breakage of one part, of this chromosome, it gets stuck to this chromosome. Okay, so it is a like translocation, it is getting translocated. Okay, it is getting translocated. So translocation may be of two types. One is called as reciprocal translocation, and second one is called as Robertsonian translocation. Reciprocal and your Robertsonian translocation. Okay, so let's discuss the reciprocal translocation. It is generally involves the breakage of two non-homologous chromosomes. Okay, means the centromere is spared. Okay, so it is the let's discuss about reciprocal translocation. Okay, it is basically the breakage of two non-homologous chromosomes, okay, in which the centromere is paired. Okay, the centromere is paired and the broken segments are just exchanged. Okay, there is no loss or gain of gene. Okay, no loss or gain of any gene. Okay, just they are exchanged. Understood? So this the individual may not show any effect as such. Okay, let's say here we have a chromosome. Okay, let's draw it in this way. Okay, let's draw it in this way. Here is one and another one I'm drawing in pink. Here is one. Okay, so what happens, this breaks. Okay, and this gets joined here, so it will look like this something. Okay, and here also what will happen, this will break and will join here. Okay, this will look like this something. It's like this, and a pink segment here like this. Okay, and the pink one will look like this and a blue segment here. Sorry. And a blue segment here. Okay, this is called as reciprocal translocation. Understood? Reciprocal translocation. And then we have the Robertsonian translocation. Okay, Robertsonian translocation. So about Robertsonian translocation, let me tell you something. Okay, so Robertsonian translocation involves two acrocentric chromosomes of different homologous groups. Okay, this involves two acrocentric chromosomes of different homologous groups. Okay, so centromere is also broken. There are two breaks at centromeres. Okay, this will cause the breakage of a short and long arm of the two chromosomes. Okay, and this will cause the trisomy of that chromosome. So for example, let us understand by diagrams. You want to understand the language. Okay, between acrocentric chromosomes of 14 and 21. Okay, so this is a Robertsonian translocation. We write this as 14q to 21q translocation. Okay, this is the notation in which you write. Let us discuss this. Like one chromosome is there like this. This is one. Okay, and another one is there. This is one. Just assume that this is your chromosome 14. What will occur is that this part, okay, this part will go here and this part will go here. Okay, so this will become like this, chromosome 14 and 21. Okay, this will become like this. This and this. This and this. Okay, understood. So this is called as a Robertsonian translocation. Understood? Okay. Okay, so after this translocation, let's discuss about a Cri-du-Chat syndrome or Cri-du-Chat syndrome. Okay, Cri-du-Chat syndrome. Okay, about the Cri-du-Chat syndrome, or it is called as a cat's cry syndrome. What are the silent features of the cat's cry syndrome or the Cri-du-Chat syndrome? Let me show by a, if I have a diagram, let me find it. Okay, so here you can see some features of Cri-du-Chat syndrome. Let us look at the features later. So this is a structural abnormality. This is a structural chromosomal abnormality or aberration in which there is a deletion of your deletion of terminal portion of short arm of chromosome 5p minus. It is also written as this. Okay, so what are the clinical features? Okay, so you can see round face. The face is round. The ears are low set. Okay, low set ears you can observe in this patient. Okay, you can observe in the subject that the subject has low set ears. Okay, microcephaly, the brain development is retarded. Severe developmental delays have occurred. Okay, severe developmental delays and cognitive deficits. It is due to your microcephaly only. And facial abnormalities also, like hypoplastic nasal bridge, the low set nasal bridge or hypoplastic nasal bridge. So you can write clinical features. Okay, this is called cat's cry syndrome. Cri-du-Chat means cat's cry. It is a shrill cry. The infant has a shrill cry or cat-like cry. Okay, microcephaly, short brain, small brain, microcephaly. Okay, another thing is round face, and patient has oblique palpebral fissure, oblique Look at this. The fissures are just oblique, palpebral oblique palpebral fissure, which is called hypertelorism. Hypertelorism. Okay. And another thing is saddle shaped nose, saddle nose. Okay. Mental retardation. And graying of hairs. Graying of hair. Okay. So this is about your Cri-du-Chat syndrome. So if you get a short question, a short note on Cri-du-Chat syndrome, you can write well. Okay. Then let's talk about another syndrome. Okay, which is called as your infamous Down syndrome. Down syndrome. Okay, so Down syndrome is an important part. You may get a short question on this. Okay, so let's start with the Down syndrome. So Down syndrome, you can see the patient here. Okay, some features are there which can be seen. Okay, let's not go to features first. Let's get the introduction. It is the most common numerical abnormality. The Cri-du-Chat was structural abnormality. This is the most common numerical abnormality. Okay, numerical abnormality of the chromosomes. Okay, it was first described by Langdon Down, described by Langdon Down in 1886. Okay, the frequency is about 650 to 700, not 6500, 700 per live births. Okay, this is the frequency of occurrence of your Down syndrome. Okay, Down syndrome in the genotype, about the genotypic changes, about the genotype. Okay, it is called as a trisomy 21. Okay, trisomy 21. So genotype is that 47 XX plus 21 or your 47 XY plus 21. Okay, this is your, depending upon male or female, this is your genotype in the Down syndrome. Okay, what is the risk factors? What are the risk factors for Down syndrome? Okay, so the risk factors are generally not this doesn't occur in mainly adults. It is increase in age of mother at conception. Okay, like above mothers above 35, more than 35 years of age, 35 years. Okay, they give birth to say babies with Down syndrome. Okay, babies with Down syndrome. Okay, so what are the clinical features? Clinical features are important here. You can observe some clinical features like your unplanted palpebral fissure. Okay, upslanted palpebral fissure. These are like upslanted. Okay, flat nasal bridge is there. Okay, nuchal folds can be seen in your cervical region. The nuchal folds can be seen in your cervical region. Clinodactyly of your fifth finger. Single palmar flexion crease is there, which is called as a simian crease. Okay, and hypotonia, the loss of muscle tone is there. Okay, what is the clinical features? So let me write it. So round face with oblique palpebral fissure and upward slanting. Okay, it is called as a mongoloid face. Okay, so the important thing is to write mongoloid. Mongoloid face. How will you describe a mongoloid face? It is a round face. With oblique palpebral fissure and upward slanting of it. Okay, the palpebral fissure will slant upwards. Okay, then you have flat nasal bridge, flat nasal bridge. Then you have low set ears, same feature as Credo chat, low set ears. Okay, you have low set ears. Then you have open mouth with protruding tongue. You can have open mouth, thick tongue or protruding tongue is seen here. Okay, then you have single palm crease, single palm crease, single palm crease, which is called as a simian crease. Simian crease. Okay, simian crease is a characteristic feature. Then you have short stature. Okay, short stature, and you have the clinodactyly of fifth digit. Okay, so fifth digit is curved inwards. Okay, which is called as a clinodactyly. You can write a host of features on the Down syndrome. Okay, after the Down syndrome, this is about your Down syndrome, just look at this. And after the Down syndrome, let's discuss the Klinefelter syndrome. Okay, let's discuss the Klinefelter syndrome. So if I am discussing the Klinefelter syndrome, okay, so just look at the features of the Klinefelter syndrome. Okay, so you can observe a real patient here having the Klinefelter syndrome. Okay, it is also due to a numerical abnormality. So Klinefelter syndrome is XXY condition, which is also a numerical abnormality. Okay, or aberration of chromosomes. Okay. The cause is due to non-disjunction. Of XX chromosomes during oogenesis. Okay, there is absent disjunction of XX chromosome during oogenesis, due to which a extra X is able to stick to this. Okay, in which the bar bodies in your Klinefelter syndrome will be 2 minus 1, that is 1 bar body. Okay, bar body is 1. Understood? It was described by Klinefelter in 1942, and the frequency will be 1:500 male births. Okay, 1:500 male births. Understood? What is the genotype? So genotype I told already. Let us state this. So genotype is 47XXY with bar body present. Okay, and the characteristic features, the clinical features will be tall individual with long arms and legs. Okay, so just observe this. We'll discuss the features here. Tall individual. Okay, long arms and legs. Okay, then gynecomastia. See, the breast development is occurring here. Okay, so the individual has gynecomastia also. Okay, and if you just focus on the testis, okay, then you will find small testis and azoospermia. You will find small testis because there is one extra X chromosome adding to the femininity. Okay, small testis with azoospermia. Infertility may be there, azoospermia and infertility. And hypospadias may be present. Hypospadias means the opening of the penis, the penile opening is present on any other location except the tip of the glans penis. Okay, so this is called hypospadias. So these all are the clinical features of the patient. Okay, so this is Klinefelter syndrome. Okay, now let's discuss about the another syndrome, that is your Turner's syndrome. Turner's syndrome. So if I go on discussing about the Turner's syndrome, so Turner's syndrome is also a numerical anomaly of aberration of chromosome. This is also numerical chromosomal abnormality. Okay, the loss of X chromosome is there. The genotype is XO. Loss of X chromosome is there. Okay, it was first described by Turner in 1938. The frequency is about 2:3,000 female births. Okay, understood? About the genotype, I told it is 45XO. Understood? And the phenotype is a female with absent bar body. Okay, let's look at a female with Turner syndrome. Okay, so here is our patient, a girl with the Turner syndrome. Okay, and what is the features, we'll discuss it here. Okay, you can see she has short stature. Okay, short stature, the height is short. Okay, and with webbed neck. Look at the neck portion, it is like webbed and wide. Okay, so webbed neck, and again sealed chest with pinpoint nipples. Sealed chest with pinpoint nipples. Okay, the chest is sealed like, with the nipples are pinpoint. Okay, they're just protruding out. Okay, and infantile genitalia or infertility must be there. Okay, infertility, because there is loss of femininity. And bilateral cubital valgus will be there. Okay, cubital valgus, the hands are abnormally bent. Okay, so bilateral cubital valgus. Okay, this will be there. And there will be amenorrhea. Understood? So after this we finish our syndromes. Turner syndrome, after Turner syndrome we finish it. Let's write sometimes you may get about something, they may ask you something about sex linked genes. So first thing that you must note a fact, that Y chromosome is shorter than X chromosome. Okay, and the genes on sex chromosomes on X or Y are what type of genes? I told you earlier, these are called as sex linked genes. Okay, so depending upon the sex linked genes, inheritance can be X linked inheritance, or your Y linked inheritance. Okay, so the disorders which are labeled as Y linked inheritance disorders must be only in males, because Y chromosome is present only in males. Understood? For example, your hairy pinna Okay, this is a disorder of your X-linked inheritance. Okay, this may be dominant or recessive. The X-linked inheritance is primarily observed in males, but it can be observed in females also. But Y-linked inheritance is also observed in males. Sorry. Okay, so in X-linked dominant inheritance. Okay, so we have X-linked dominant trait, X-linked dominant, and X-linked recessive. X-linked recessive. Okay, the X-linked dominant traits, we have three diseases, okay, which are X-linked dominant. Here you can remember three diseases which are, okay, so X-linked dominant, we have Rett syndrome. Okay, we can have lissencephaly, and we can have hypophosphatemia. Okay, you can write examples of X-linked dominant. Okay, just remember these three examples. Okay, so the affected male, the X-linked dominant trait is there. Okay, so the affected male, so the male will be having X chromosome. Okay, XY, this is the man. Okay, there is the problem here, so I'm marking it with asterisk sign. Okay, and the female, let the female be normal. Okay, the female is normal, XX. Okay, let the female be normal. Okay, if they produce a girl, the male will obviously donate X chromosome. Okay, and if they produce a boy, the male will donate his Y chromosome. Okay, so the male will transmit the disease to his daughters only. Okay, to his daughters. Okay, the male will transmit the disease to his daughters. So affected heterozygous females will pass the disease half to sons and half the daughters. Okay, man will transmit disease to daughters if the man is affected. Okay, this is the case of affected man. Okay, if there is a case of affected the wife. Okay, the wife is affected, cross XX. Let's say the wife is affected. Wife is affected means both must be there, otherwise they won't be affected. Let's say this both are defective. Okay, if they produce same boy, okay, so XX affect, and if they produce a girl, this will be their XX is girl now. If they produce a boy, then XY. Okay, XY. Okay, XY. Understood? As this is a X-linked dominant trait, so this is considered your diseased. Okay, this is considered your diseased here. Okay, and normal may be also produced by this combination, and normal. Okay, so the women will the affected women who is affected will have transmitted the trait to half of sons and daughters. Okay, this have transmitted it to half the sons and daughters. Understood what is the inheritance? So this is the X-linked dominant inheritance pattern. Okay, understood? And then we will discuss about a X-linked recessive inheritance pattern. Okay, then we have disorders like X-linked recessive. You can have hemophilia. Which is called as your bleeder's disease. Okay, bleeder's disease or a royal disease. Then we have color blindness, and then we have Duchenne muscular dystrophy. Understood? Dystrophy, DMD. You can remember these three examples for X-linked recessive disorders. Okay, okay. So if you just want to know about the inheritance, okay, the inheritance followed by the X-linked recessive is called as a zigzag inheritance. zigzag inheritance pattern. Okay, I'm not going details, in these topics, because if I go into the pedigree analysis part, okay, if I tell you that this into your affected this, okay, is affected, they produce some offsprings like this, and half of this is colored shaded, okay, and they produce some consanguineous marriage is there, okay. The pedigree analysis can be complicated in a variety of ways, okay. So in your textbook, that is your gold standard textbook, your MREs, your MREs pedigree analysis are focused mainly, okay. So I'm not going into that depth, but I'm just teaching very very superficially of genetics, or the introduction of genetics, medical genetics, okay. So which will be helpful for only your prof and some extent to your PG, okay. Okay, so it is showing zigzag inheritance, okay, that the female, if the female are the carriers and the male are the sufferers here, the female are the carriers and the males are the sufferers. The males are the sufferers, okay, understood. So what do you mean by zigzag inheritance? Zigzag inheritance means that it escapes one generation and trait appears again in the next generation, okay. It generally the trait disappears within the generation, okay. So this is just the inheritance pattern, okay, inheritance pattern, okay. If you want to know any other thing, okay, if you want to just know a disorder that is based on autosomal dominant inheritance, okay, and autosomal recessive inheritance, so we have autosomal disorders also. So we can have autosomal dominant. We'll discuss one example of this. And we can have autosomal recessive also. Okay, so autosomal dominant, we can have example your Marfan syndrome. So what I am teaching is strictly within the syllabus, within your syllabus, not beyond that, okay. And autosomal recessive is your phenylketonuria, okay, phenylketonuria. So Marfan syndrome, it is an inherited disorder, okay. Let's look at a patient. If I have just, okay, so here's the picture. This is the Marfan syndrome. In Marfan syndrome, it is an inherited disorder which affects generally the connective tissues, okay. So it affects generally the connective tissues affected, connective tissues are affected, and it is caused by autosomal dominant inheritance, okay. Like one defective gene from either parent can cause it, okay. About 25% of cases, mutation in FBN1 gene, okay, FB1 gene generally helps in the codes for your fibrillin protein, okay, protein called fibrillin, which is essential for synthesis of connective tissues, okay, fibrillin, okay, this is essential for connective tissue, okay. So mutation FBN1 gene causes it in about 25% of cases. So what are the clinical features? About the clinical features of a Marfan syndrome, okay. The first one is disproportionately long, the proportionately long legs and feet, okay, long extremities. But the only thing is it is within proportion, okay. The proportion is like hands are long and the legs are also long, as in as a normal human being has in proportion, okay. Then slender build, you can see the girl is very slender build, okay. Then we have arm span is greater than height. Okay, arm span is greater than height. Okay, this is just an exaggeration. You can have long spidery fingers or arachnodactyly. Arachnodactyly. What is arachnodactyly? So it is, we have long fingers. You can see long fingers. Okay, this is called as arachnodactyly. Okay, then what can you have? You have crowded teeth. Okay, crowded teeth. Ectopia lentis. Ectopia lentis. Ectopia lentis. Ectopia lentis is a deviation of the lens towards one side. Okay, and another very important feature is the funnel shaped chest. Okay, chest is compressed anterior posteriorly. So you have funnel shaped chest, funnel chest, which is also called as a pectus excavatum. Pectus excavatum. Okay, pectus excavatum. And you have aortic dilatation also. You have aortic dilatation also. Understood? Then we will talk about the phenylketonuria. Phenylketonuria is an inborn error of metabolism. Inborn error of metabolism. Okay, autosomal recessive in inheritance. Okay, so the enzyme, okay, the enzyme which the gene responsible for producing the enzyme, which enzyme is absent? Absence of phenylalanine hydroxylase. Phenylalanine. I believe you must have read about phenylketonuria in very detail in your biochemistry classes. Hydroxylase. I'm not going into deep about phenylalanine, how it is. This phenylalanine. Okay, this is converted into phenyl pyruvate and your phenyl lactate. Okay, phenyl pyruvate, phenyl lactate. Okay, and your urine is urine is called as a Mosey order of urine. Okay, Mosey order of urine. These facts are biochemical significant facts. Okay, so I'm not going into much detailing. Okay, it is as it is recessive trait, it is only homozygous. Okay, it is a trait which is expressed only in homozygous state. Okay, it is common in marriage between relatives, common in consanguineous marriage. Consanguineous marriages. Okay, consanguineous marriages is marriage between relatives. Relatives or close members. Okay, close members of your family. Okay, so clinical features. In the clinical features, phenylketonuria is a host of clinical features in biochemistry. Okay, there is decreased pigmentation, depigmentation, and due to accumulation of a phenylalanine in your brain, you have mental retardedness. Or mental retardation. Okay, I wrote metal, right? Okay, mental retardation, and if a host of properties which you read in biochemistry. Okay, so refer biochemistry for details. Done. So here we complete our whole genetics. Okay, the genetics which is required for your anatomy is now completed. This much genetics you have to read and understand. Okay, these things you have to correlate, and the disorders are mainly important. Your Down syndrome, your cre du chat, your Klinefelter, your Turner syndrome, okay, this phenylketonuria, Marfan syndrome, Lyon's hypothesis, and your types of genes and bar body about short note, okay, chromosome short note, karyotyping short notes. 1 and 2 short notes are such important, not much important here, okay. But genetics as a whole, okay, it is very important for your MBBS career as well. You will not encounter any cases in clinics much, but you must have knowledge so as to do research on these topics also, okay. So you can read more from MREs or your GK Pal genetics. That's all which is required for your Prof and PG, okay. Thank you and have a nice day ahead, okay.